Assalamualaikum warahmatullahi wabarakatuh. My name is Resky Ramadhan from the group 14. I will present material on animal biotechnology. Okay, let's just go to material. Animal biotechnology. What is animal biotechnology? Animal biotechnology is the use of engineering and science to create modified living organisms to improve animal and develop something agricultural cultural and pharmaceutical application. Animal biotechnology includes all animals, livestock, poultry, fish, insect, companion animals, and laboratory animals. History of animal biotechnology. The animal biotechnology in use today is built on a long history. Some of the first biotechnology in use includes traditional breeding technique that date back to 5000 BCA. Such technique include crossing diverse strains of animal, known as hybridizing, to produce greater genetic variety. The offspring from these cross then are bred selective to produce the greatest number of desirable traits. For example, female horses have been bred with male donkeys to produce mules, and male horses have been bred with female donkeys to produce hinnies for use as work animals. For the past 3000 years, this method continues to be used today. The modern era of biotechnology began in 1953 when American biochemist James Watson and British biophysicist Francis Crick present their double helix model of DNA. That was followed by Swiss microbiologist Werner Erber discovery in the 1916 of special enzymes called restriction enzymes in bacteria. These enzymes cut the DNA strands of any organisms at process points. In 1973, American genetic Stanley Cohen and American biochemist Herbert Boyer removed a specific gene from one bacterium and insert it to it into another using restriction enzymes. The, the event marked the beginning of recombinant DNA technology or genetic engineering. In 1977, genes from other organisms were transferred to bacteria, and achievement that, that led eventually to the transfer of a human gene. Animal biotechnology sector. Application development through research have led to the emergence of true scientific agricultural animal biotechnology sector. The first, animal genomics. Genomics defines and characterizes the complex genetic makeup of an animal. By understanding the genomes of animals, we can better understand the basis for disease resistance, disease susceptibility, weight gain, and determinants of nutritional value. The second, animal cloning. Using somatic cell nuclear transfer, livestock breeders can create an exact genetic copy of an existing animal, essentially an identical twin. Cloning does not manipulate the animal genetic makeup nor change an animal DNA. It is simply another form of sophisticated assisted reproduction. And the last, genetic engineering of animals. Manual manipulation of DNA to change the characteristic of an organism using biotechnology. This means changing the genetic makeup of the cell by removing and editing DNA or genes to enhance the organisms and go beyond its limitation. It can also mean taking the DNA of another organism and incorporating it into the DNA of the recipient organism, thereby adding new traits not previously present in the receiving organism. Next, advantage of the application of animal biotechnology. Combin- Combining animal and biotechnology result in advance in four primary areas. The first, animal biotechnology to advance human health. Animals have been used for years to produce medicines for humans. Animal-made pharmaceutical transfer biotech animal into factories to produce therapeutic proteins in their milk, eggs, and blood, which can be used in the development of biopharmaceuticals. In addition, biotechnology can be used to produce human-compatible transplant organs, tissues, and cells in pigs that can be vital to enhancing human health. The second, biotechnology to improve animal health. For decades, farmers have been improving livestock health through enhanced animal husbandry practice and more modern technologies such as artificial insemination, embryo transfer, in vitro fertilization, genetic mapping, and cloning. Through biotechnology, farmers can enhance breeding, resulting in healthier herd. Additionally, the animal health industry has developed treatments that can prevent and treat disease. New vaccine, diagnostic tests, and practice can help farmers treat animal disease while reducing foodborne pathogens at the farm level. Third, biotechnology to develop more nutritious food. Improve animal health conditions from vaccine, medicine, and diagnostic tests result in safer foods for consumers. In addition, food quality may be improved by introducing desirable traits through new gains into farm livestock and poultry. In the future, meat, milk, and egg products from animals can be nutritionally enriched with the use of biotechnology. And the last conversation of earth, environment, and animal. Biotechnology can help produce environmentally friendly animals as well as conserve endangered species. Farm animals and their feed have been improved through biotechnology to reduce animal waste, minimizing, minimizing the impact on the environment. Today, reproductive and cloning techniques offer the possibility of preserving the genetic and endangered species. Genetic studies of endangered animals can also result in increased genetic diversity, which can result in healthier population of species. Thanks for attention. Assalamualaikum warahmatullahi wabarakatuh.